So today the topic it's Autophagy 2.0. And my guest again is Professor David Rubinsztein from University of Cambridge in England, where he's a professor of molecular genetics. He's kind of a major contributor to the understanding of potential for alterations in autophagy, impairment in autophagy, and the accumulation of proteins that are in neurons in Alzheimer's, Parkinson's, ALS, and Huntington's diseases. And so if you viewers and listeners remember last time, Professor Rubinsztein talked about kind of what is autophagy. It's the shuttling of garbage, dysfunctional damaged proteins, organelles like mitochondria to the acid bath of lysosomes. And as he'll talk about today, there's a lot of accumulating evidence, both from standpoint of genetics and then experimental manipulations in experimental models, that impaired autophagy is contributing, maybe very importantly, to the demise of neurons in these different disorders I mentioned. So welcome back, David.
Nice to see you, Mark. Thanks for inviting me.
Yeah. And I think maybe to start with, why don't you and I maybe do like a tag team thing and describe, so Alzheimer's, Parkinson's, ALS, Huntington's disease, but just what does the brain look like, or in ALS, the spinal cord? What are the proteins that are accumulating in, or in the case of Alzheimer's, also outside of neurons? And then get into why autophagy seems like a very important mechanism to remove these proteins. So I guess I'll start with Alzheimer's disease. Most cases of Alzheimer's, we don't know the cause. We know that advancing age is the major risk factor, so don't get old. No. And so that's a big problem, because by the time we get, David, myself, everybody listening to this, when we get into our 70s and 80s, there's a real chance that we're going to end up with dementia before we die. And it's not an insignificant, it's up to 30-40% chance by the time we reach 80s. So we're all at risk. And so it's important to understand what's going on with aging that's apparently contributing to the disease. And then is there anything we can do to reduce our risk, which most of us in these fields now are working on these neurodegenerative disorders. Some of us anyway, myself and others, David, I think, are kind of shifting to, are there treatments we can give to someone when they're already symptomatic that can reverse the disease? So someone who's having learning and memory problems, short-term memory problems, is there some drug or some manipulation we can do, and all of a sudden they're back to normal? And I don't think so. I think David's on the same wavelength. So last thing with Alzheimer's, if you look at the brain, so it's very clear there's accumulation of a protein called tau inside of the neurons that are degenerating. And that tau is abnormal in the sense that there's what we call a post-translational modification, addition of phosphates to the proteins called phosphorylation. And this tau protein, its normal function is in the control of microtubules, these polymers in neurons. Professor Rubinsztein talked last time about how the autophagosomes, which kind of surround and enclose the garbage, are moved, in the case of neurons, apparently from way out in an axon back to the lysosome, the acid bath in the cell body. So we know just from that simple thing, the normal function of tau is in the control of the polymerization state of these microtubules. We also know that it's phosphorylation of tau can promote the depolymerization of the microtubules. So, and this is something, that I somehow kind of got lost. A lot of people don't talk about this, but when this tau is hyperphosphorylated, it comes off the microtubules and they tend to depolymerize, which, geez, that should mess up autophagy right there because things aren't moving back. But anyway, this tau accumulates in the neurofibrillary tangles. Then the other issue in Alzheimer's, that's a diagnostic thing when you look at the brain, initially for a long time at autopsy, but now this can be done actually in living humans essentially you can do amyloid imaging to look at the amyloid beta peptide accumulation outside the neurons. So you got the amyloid accumulating outside and on the surface of the neurons, which causes problems. For example, we showed it causes oxidative stress in the membrane and so on. Okay, so that's Alzheimer's disease. There's no treatments that are effective in slowing the disease process. There are few treatments that have a modest, very modest effect in kind of enhancing attention and memory. So that's the state with Alzheimer's. So it's your turn on the tag team here. Why don't you do Parkinson's?
Yeah. So maybe I'll try to sort of encapsulate the whole lot, if that's okay, Mark.
Oh, okay.
So I mean, you mentioned in Alzheimer's disease, the two pure proteins that accumulate are amyloid beta and tau. And there are data from a variety of angles that argue that the tau is a very important driver of pathology in Alzheimer's disease. Indeed, mutations in tau can give you a different form of dementia with Parkinsonism. Yeah. There's an accumulation of proteins also in Huntington's disease, in motor neuron disease, and in various other neurodegenerative diseases. And for instance, various spinocerebellar ataxias as examples. In these cases, when the protein accumulates, the proteins that accumulate inside the cells are not simply accumulating, they're also forming clumps, aggregates inside the cells. And although there's a debate about which species and which aggregates are necessarily bad, I think there's consensus that the propensity of these proteins to form aggregates is a characteristic that is associated with their deleterious properties. And to put it sort of in genetic speak, simplified, these proteins are poisons for the cell. And for all of these examples that we mentioned, there are good data, at least in animal models, that if one were to be able to lower the levels of tau or Huntington in Huntington's disease, or the protein that drives Parkinson's disease, in most cases alpha-synuclein, that this is likely to be protected. Okay, so just to, and I should say in Parkinson's disease, we know that alpha-synuclein, the protein that is accumulating, can be expressed at abnormally high levels in certain rare genetic forms of Parkinson's disease where people have extra copies of the gene.
Yeah.
And so that argues that for Parkinson's disease, that we know that overexpression is sufficient to cause disease.
And that was Andy Singleton, I think, who had the first family where there's a triplication of the gene. So essentially there's a 50% increase in the amount of the alpha-synuclein, and that alone, that was very important, like you're saying, that's sufficient. But that also, David, doesn't that suggest that if autophagy is what's normally removing these aggregates or these clumps, that it's like in a very delicate balance where just a little bit too much of the protein starts to aggregate, and then this autophagy isn't able to deal with it. Is that absolutely?
So so I think you're absolutely right, Mark, but maybe I should rewind to sort of
Okay.
try to elucidate the logical steps.
Okay.
So that you've got already. Okay, so I think the first point is that these diseases are generally caused by proteins that tend to aggregate, and these proteins are poisonous for the cell. So these are mutations that cause diseases predominantly because you've lost activity. They cause diseases because they're poisonous, they're toxins.
Yeah.
And we showed about 20 years ago that autophagy was a route for removing these proteins. And so if we blocked autophagy at different stages, we increased the levels of these proteins for tau, for Huntington, for alpha-synuclein, and made things worse in various disease models. And importantly, we showed that if we boosted the formation of autophagosomes, we could attenuate the disease by lowering the levels of these proteins in models from cells to Drosophila to zebrafish to mice. So these suggest two things. Firstly, that autophagy is an important mechanism for keeping the levels of these proteins under control and stop the poisons building up in the cells. The second is that there's a potential therapeutic mechanism which could boost autophagy. So you asked the question whether there's a delicate balance, and there's a very delicate balance, because if autophagy is compromised, then these proteins are going to accumulate and things are going to get worse. But it's actually worse than that, because we've shown, for instance, that Huntington and alpha-synuclein excess, mutant Huntington and alpha-synuclein excess in a cell-autonomous manner, so without any influence of other cells, which is something I'll come back to, in a cell-autonomous manner, impair autophagy. And so you've got a situation where the mutant protein has the potential to impair autophagy, but the mutant protein is itself removed by autophagy. And so there's the potential for a positive feedback loop.
Yes.
And so you talked about aging, and clearly aging is the major risk factor for many of these diseases. And we've shown fairly recently, supporting ideas from others, but we've shown it specifically in the brain, that autophagy decreases in the mammalian brain. We did it in mice with age. And as a consequence, you might have a situation where you have an organism or person with the propensity towards one of these diseases. And what happens is that they keep things in check. Okay, either the autophagy copes or other protein degradation systems can kick in and help. So there's the ubiquitin proteasome system as an example, which sort of supports things. But you can envisage a situation where at a particular age, the ability of the cell to buffer the accumulation of these toxic proteins, or one of these toxic proteins, gets compromised, potentially as a consequence of autophagy rates falling off with age. And when that happens, you can get a buildup of the protein, you get further compromise of autophagy, further buildup of the protein, and even greater compromise of autophagy. And I think that could lead to, in a cell-autonomous manner, a very rapid accumulation of the poison in the cell. And although this is a very simplistic way of thinking about it, I think given this relationship between the proteins being autophagy substrates and the proteins themselves often being inhibitors of autophagy, there's this potential for things to go bad really quickly once the homeostasis starts creaking. So I have a sort of captured what you were thinking about.
Yeah, and one thing we don't really know clearly is what's the time course of death of individual neurons in the brain, because these neurofibrillary tangles are mostly dead neurons. I think they're kind of the remnants And so when you were talking there, I was thinking, you start accumulate these toxic proteins, and then you got this feedback loop where autophagy can't handle it. That leads to more of the toxic protein. And, so the neurons could actually die, go from alive to dead, individual ones, pretty quickly. We don't know, I don't think. It could be, even days, I don't know. There's this process of apoptosis, right? And then more recently, this ferroptosis, which has to do with oxidative stress and iron. And in those situations, it's like the cell gets to a certain point, and then this machinery to die is activated, and the cell dies pretty rapidly. So
I don't think people understand, what the relationships are. I mean, people done experiments. I mean, Steve Thigpen has done very elegant live cell imaging experiments, and clearly cells can form aggregates and still be alive.
Yes.
For Huntington. But I think that, knowing these dynamics and knowing how functional a cell is at different stages. So cells might be sort of in very bad shape and still alive and not much use, or actually deleterious. I mean, can I wind back? Sorry, just a sec. I think there's one other point that's important to make is, of course, autophagy affects many other processes. So defective autophagy, while it would lead to the accumulation of these poisonous proteins, can have many other effects that are not going to be good for neurons or even glia. I think the other point to make is these poisonous proteins have pretty promiscuous toxicity. They hit many different systems. And so, I think that when we're thinking about therapeutics for these diseases, if we look at specific downstream steps, that could be tricky because we don't know which are going to be rate limiting in a person. And I think that that's why a focus on trying to limit the levels of the poison is a very rational strategy.
Yes. And okay, so these proteins, like with tau, for example, in Alzheimer's, and you mentioned the frontotemporal dementia, which are caused by mutations in tau, so tau is normally in the axon. Like if you stain healthy neurons with an antibody to tau, it's in the axon. It's not in the dendrites or the cell body. But then in Alzheimer's, it's accumulating in the cell body. And so, and last time we talked briefly about Randy Nixon's work too, where, he's looking at the accumulation of autophagosomes, which indicates there's a problem in getting rid of the garbage. And now I can't remember, does he see the autophagosomes accumulating only in the cell body or also out in say axons or dendrites?
Yeah. So Randy's data suggests that in Alzheimer's disease, there's a primary defect at the level of the lysosomes.
Yes.
And so essentially the cellular incinerators are not working. And when that occurs, you get an accumulation of autophagosomes. The accumulation of autophagosomes looks like it's occurring actually in the axons. A lot's occurring in the axons. And in fact, the so-called dystrophic neurites might contain many, how can I say, non-digested, not properly digested autophagosomes. So that might be an important part of the pathology. And actually, just sort of to reiterate its work, which I think, raises important hypotheses. As you mentioned, the amyloid beta that is seen in Alzheimer's brains looks like it's outside the cells. But Randy's data suggests that actually originates from amyloid beta that's delivered to the lysosomes that are not functional. And then the lysosomes cause a form of cell death. And what is left is the corpse of the cell. It's like the skeleton, the amyloid beta. And so it hasn't been necessarily secreted. It's just what's left. It's the relatively insoluble material that's just left. And I think that's, to reiterate what we said last time, I think that that's an important line of investigation to follow, because it might harbor important clues about both pathogenesis and therapeutic opportunities.
And it could also, so last time we talked, I briefly mentioned this, that cells can spit out apparently toxic things, maybe protein aggregates. So, and in the scenario you just mentioned, the amyloid could also be spit out in these exosome vesicles.
Yeah, I'm not sure that he's looked at that, but
we looked.
Others, and we've looked at some stuff. And if you poison the lysosomes, you tend to increase the unconventional secretion, and you could certainly get that tau, for instance.
Yeah, because my colleague at NIA, who was actually under me there, Dimitros Kapagiannis, has done a lot of work showing, and we helped him with some cell culture stuff too, that bottom line is neurons can spit out both A beta and tau in these extracellular vesicles. Okay, so then the so Parkinson's alpha-synucleins accumulating inside neurons, Alzheimer's, tau, and maybe amyloid beta peptide as a result of dysfunctional lysosomes, and then synuclein in Parkinson's and Huntington. And then in ALS, which we really haven't mentioned yet, this is an interesting story, because back in the early 90s, we were working on oxidative stress and its potential role in age-related neurodegeneration, Alzheimer's, and then Brown up at Harvard, he found a family where there's mutations in gene encoding an antioxidant enzyme called superoxide dismutase 1, SOD1. And that protein accumulates in neurons. And initially the thinking actually, the mutations in SOD1, it must be the antioxidant enzyme isn't functioning. Therefore, the neurons can't deal with oxidative stress and they degenerate. But it turns out that's not true. There's the enzyme still functioning. Is what we call gain of function, and this protein, much as synuclein, tau, Huntington, is accumulating inside the neuron. But then more recently, there's other work there, like with Brian Traynor, who was under Andy Singleton, describing this C9orf72 gene. But okay, so why don't we switch now and talk about the genetics? Your training is in, as a geneticist, molecular geneticist. So maybe it's a good time to talk about, okay, so there's this evidence from you and others that impaired autophagy at one level or another, could be lysosome dysfunction or maybe some other problem is contributing to the accumulation of these poisonous proteins in the neurons. Are there any genetic mutations, either where the mutations in genes that encode proteins that are involved, that are known to be important in this autophagy process? Because one might predict there could be some neurological disorders that are related to abnormalities in those genes.
Yeah, that's an excellent question. So people had made mouse knockouts of core autophagy genes or specific neuronal knockouts of those core autophagy genes, and the mice get neurodegeneration, and they get the accumulation of aggregate-prone proteins. Subsequently, actually, people have been identified with mutations in a number of core autophagy genes. And it's, I think, a sufficient number that, one can exclude the argument that the gene might be having another function. And what is very interesting about these mutations is that the affected individuals present initially with a neurodevelopmental phenotype. And there's not enough data to allow one to assess the extent to which there is a progression. But there's a hint from some of the literature that there is a likelihood that there's some progression. But I think, more needs to be done in that space. So this suggests that mutations in core autophagy genes actually affect neurodevelopment in people. It's interesting that that hasn't been seen in mice. It's possible that, some of the phenotypes that one would ascertain in people with neurodevelopmental delay are too subtle for the standard sort of batteries that people would employ in mice. What's interesting for me also is that mutations in the other core protein degradation system, the ubiquitin proteasome, so mutation in proteasome subunits, again in mice give you a neurodegenerative phenotype. But in people, they again give you a neurodevelopmental phenotype. And so I think that this is telling us something about the role of these processes in development.
Yeah.
It also suggests that in normal individuals, in normal people, the loss of the autophagy genes or significant loss of function, it might not always be absolute loss of function. A significant loss of function doesn't give you a very rapid neurodegenerative phenotype, because that is not normally seen. And sort of raises the question then whether in the neurodegenerative diseases is that you've got two things going on. You don't only have the loss of autophagy, but you also have the accumulation of this poisonous protein. And so in a sense, the neuron is dealing with two rather promiscuous toxic events, but they synergistic because they both affect each other in many cases.
Yeah.
That issue of the neurodevelopmental versus the neurodegenerative in the humans with the mutations, I don't think has been completely resolved. But those are my hypotheses and speculations.
And so when people have knocked out some of these genes that encode proteins that are involved in autophagy, and they see the developmental disorder, is that with both copies of the gene knocked out, or
yeah?
And what if you just reduce by 50%?
Yeah. So the studies that have been done of those core autophagy genes in cells, the ones that I'm aware of, suggest that they're very much recessive even at the cellular level. So a 50% reduction it doesn't have any discernible effect on flux or the rate of autophagosome formation, that you really need to get the levels down to less than 10%, maybe less than 5%, to start having big effects. And so I think these are classic recessive mutations. Maybe 10%, 5% is not always the same for all mutations, but it's not, 50% makes no difference. The heterozygotes are pretty normal, they're indistinguishable from normal.
And
so it's true recessive.
Has anyone crossbred those mice with, say, alpha-synuclein mutant mice or tau mutant mice? Or is it even worth doing? I mean, you already know from your own work, right, where you inhibit autophagy in other ways, that's sufficient to promote accumulation.
So some have done those experiments. In general, when they've done those types of experiments, they make things a lot worse. We've done experiments like that in zebrafish, and it makes things a lot worse. So but that's with knockdown or knockout.
Yeah. And okay. Yeah, I want to go back to, so you have a nice, in your neuron review article, you have a nice schematic diagram with like 4 different stages of autophagy, and then different disorders, Alzheimer's, Parkinson's, ALS, and then some genes that seem to be involved in different stages, or sorry. So there are rare cases of Alzheimer's, Parkinson's that are inherited. They're caused by mutations. So Alzheimer's, presenilin 1 is the most common mutations, but also amyloid precursor protein and PD, which you've done quite a bit of work on. There's different ones, LRK2 and so on. But anyway, can you, I don't know if you want to summarize, but in other words, for example, we were talking about Randy Nixon, and we'd done this work like in the early 90s again, where we found that presenilin 1 mutations alter calcium regulation in the endoplasmic reticulum. This presenilin 1's a transmembrane protein, and we did all these experiments in culture mainly, had that evidence. And I think Randy had some evidence that the presenilin mutations affect lysosomal pH by secondary to an effect on calcium, if I remember right.
Oh, well, he showed there was defective trafficking of the ATPase that effectively acidifies the lysosome. But I think that sort of schematic has an important message in it. And I want to sort of maybe make the point, because I think that, if one takes any of these diseases, but let's say Parkinson's disease, what we try to illustrate is that there's some mutations that cause Parkinson's disease that impact the formation of autophagosomes. So as an example, if you've got excess copies of the alpha-synuclein gene, then the disease that present with Parkinsonism, where there's a defect in the dynein-dynactin complex. In that case, the autophagosomes are built properly, but they're not taken to the part of the cell where the lysosomes are. And then there are forms of Parkinsonisms or Parkinson's disease where the mutations that affect lysosomal function. I mean, glucocerebrosidase mutations are a good one, but there's a very classic example with ATP13A2, which is an ATPase regulating lysosomal function. In those cases, one wouldn't use the same autophagy therapy in the different diseases. So you might want to ameliorate the diseases caused by defective autophagosome biogenesis with strategies that increase autophagosome biogenesis. But you wouldn't necessarily want to increase autophagosome biogenesis in a disease where the defect's at the level of the lysosome, because it would be like putting more rubbish trucks on the road when the incinerators are not working, or they're closed, and you're just going to cause a traffic jam with the accumulation of autophagosomes, which itself might be deleterious to cells. And so this brings me to the idea that, with that simple example, it shows how important it is for us to think about stratifying patients when we consider therapies, because we see this with monogenic forms of these diseases. Sure as anything, that principle is likely to apply with complex forms of the disease. So in Alzheimer's disease, I'm not saying in an autophagy context, but in a general context, there are now examples where I think people who carry the ApoE4, which is a very high risk allele for Alzheimer's disease, might respond different to people who don't to certain indications. So I think, as we move forward, and given where we come now with genetics and sequencing, we really need to stratify as first principle, not as a retrospective event, as a first principle, because people who manifest with these clinical syndromes that look the same might have arrived there through rather different pathogenic routes that respond differently to different interventions.
Yeah, so you're talking about personalized medicine approaches there. And one problem with clinical trials in humans is we're very heterogeneous in our genes. and so that, as you said, one person may have, because of some genetic variants, increased risk of impairment of autophagy at this step versus this step. And so you're kind of envisioning in the future, the more we understand, the more we can do, right? So, but with clinical trials, maybe in one contributor in some instances, maybe many instances, is that a treatment could be effective that was developed initially in an animal model or something. It could be effective or have some beneficial effect in a subset of people, but because the clinical trials, you throw them all together, and then that would increase the variability, and the drug could actually help 10% of the people, 20%, but you never see it because the statistics aren't going to show it when you're lumping those people together with others where the drug has no effect.
Yeah, and I think that the two ways of, I think that's a likelihood. I don't think it's in most cases, I don't think it's going to be the exception. And so I think you've got to consider at least two rather broad strategies, which to managing. The one is to say we think these are the important variables, genetic or other variables, that might contribute to treatment response, and build that into your analytic framework that you're going to employ to look at the trial results. I mean, another way of doing it might be to say, okay, we don't know. It's so complicated, we don't actually know, because one could stratify genetically, but you might say, okay, but we don't know how the genotype is actually going to impact the treatment response through the whole pathway. Do you know what I'm trying to say?
Yeah.
So, but then, given the scenario you present, you could say, why won't we do two types of trials, or divide your trial in two, if it's a large enough trial? And then you could say, in the first trial, or the first half of the trial, you run the trial, and then you consider the genotypes, the complex genotypes of the responders versus the non-responders, and then you look to see whether those end up being predictive in the second trial, or they're specific. Do you see what I'm saying?
Yeah.
I think if one doesn't build in that possibility, we're going to have huge problems. And an additional factor that people have become sensitized to, which I think is very important, is the likelihood that the diseases might have different drivers in different ethnic groups. And so this suggests that one needs to run the trials across different ethnic groups, which really hasn't been the case in many situations in neurodegenerative diseases, but also be cognizant of the reality that they might have, because they have different ethnic groups, they'll have different genetic compositions, and that might influence their disease drivers as well as their response drivers. And so I think that, we're at a time now where we can start unraveling some of this complexity. And I'm concerned that there might have been missed signals in many trials that have been considered to be negative.
Yes, no, I agree with that. We did some work, and quite a bit of work actually, in animal models of stroke, right? And this was way back when they were doing, there was a lot of excitement, no pun intended, in the field, because there's this evidence accumulating that excitotoxicity is contributing to the death of the neurons. And so they had this drug that blocks a particular type of glutamate receptor, and did clinical trials, and they didn't see any significant effect. But there's so much, stroke is maybe in some ways even worse than the diseases we're talking about, because, okay, you got to give the treatment after the person has a stroke, right? So then how old are they? Are they male or female, which we didn't talk about? What ethnic group? What blood vessel was it? How long was the clot in the blood vessel? Usually the clot forms in the blood vessel in the brain and then kind of resolves itself, and you get what's called reperfusion. So then you look back on it and you say, how are you going to pick up anything when there's so much variability? And the quantification was essentially functional deficits. So yeah, huge problem. The animal model, it's easy. We have genetically pure mice. Back then, most all the studies were in males. I'm guilty too. But now we all do males and females. And then, so kind of rules out the genetic problem, rules out the sex issue. All the same age, the same blood vessel in the brain, the exact same blood vessel we occlude for exactly 1 hour. And we kind of get fairly reliable amount of brain damage and motor deficits. But in the humans, it's so variable, statistically, how are you going to, even if something is having a beneficial effect, how are you going to pick it out, right? But that, I think, to a less dramatic effect also applies to these, disorders we've been talking about. Yes. That we have no treatments for. And by the way, there's still no good treatments for stroke, except for physical therapy, and that kind of thing. So you talked about the possibility of targeting different steps in the autophagy process, depending on where the problem might lie. And last time we briefly talked about rapamycin and mTOR. So then if the problem, okay, I guess first question is, how is mTOR reducing autophagy? Where is it acting in these 4 different steps?
So mTOR acts likely in a number of steps of the pathway. It inhibits the formation of autophagosome by phosphorylating some key proteins that are important in that part of the process in the initiation, and diminishes their function as a consequence. But mTOR also regulates lysosomal biogenesis largely through a protein family exemplified by transcription factor EB, so I'll call it TFEB. And when TFEB's a transcription factor, so it works in the nucleus, and when it is phosphorylated, it stays in the cytoplasm, so it can't do its job. And so I think the mTOR likely regulates a number of steps of the pathway.
Okay. And so by inhibiting mTOR, rapamycin will enhance autophagy in multiple steps? Is that?
It actually,
okay.
The TFEB effect of rapamycin is pretty minimal. You've got to use a stronger inhibitor. But so the rapamycin will work predominantly at the formation step.
Okay. All right. And then are you, there have been published studies. This is how things often happen in science. You get, like, there's this explosion of studies of rapamycin treating Alzheimer's mouse models, Parkinson's mouse models, so on, and in many cases seeing beneficial effects, I think, although I'm not sure exactly how reproducible and strong it is. So are you working on other drugs? Do you have a drug development program there?
Yeah, I mean, I think it's fair to say we published, I think, maybe the first studies with rapamycin on autophagy in neurodegenerative mouse models, and many groups have seen the same. Yeah. In various ways, including industrial groups have published. I'm not that keen on rapamycin as a drug. for neurodegenerative diseases, because along the lines you were suggesting earlier, I think we need to get in very early. And so we need to be able to treat people for long periods with safe drugs. And rapamycin is a very big molecule. It doesn't get in the brain that well. So you've got to give a high peripheral, you end up with a high peripheral exposure to get some brain activity. And that comes with side effects. The worst are the wound healing ones. So people get very bad mouth ulceration, for instance. And there's some immunosuppression, which is, that's what its original clinical indication was for. So we veered away from that and tried to identify mTOR-independent drugs. And we've done that with we did some screening. We did the first screen with Stuart Schreiber's lab, and in fact, it took us nearly 10 years to identify the target of one of our best hits from that. We have also done quite a lot of repurposing, and one of the repurposed drugs, it's an L-type calcium channel blocker, which gets into the brain and induces autophagy, and in fact induces autophagy in mice at concentrations similar to those seen in people. That's in a clinical trial that is being driven largely by my colleague Roger Barker in Cambridge in early Huntington's patients. So, we have a program. I don't know if that's the word, but we have an active effort to try to identify targets and potential drugs that might hit on targets. We don't have a biotech level programming, that we don't do a lot of screening and that type of thing. We do a little bit.
Yeah. And then what about natural ways? I think briefly we talked, when you were talking about basic mechanisms of autophagy and its discovery in the yeast studies, and that led to a Nobel Prize identifying genes involved in autophagy. And we talked about the way that Japanese scientist was stimulating autophagy was by essentially starving the cells, or food deprivation, fasting, which humans can do. And so food deprivation in animals, fasting in humans will stimulate autophagy, and it inhibits mTOR. And that's a safe thing.
Yeah, so people have done studies in mice, even with food deprivation. I can't remember which neurodegenerative disease model, and it did what you'd predict. So it was
we did the first studies with that.
Okay.
Yeah.
But I think the problem is translating mouse fasting to human fasting. As I think I mentioned last time,
you
fast a mouse for 24 hours, it's like 7 days for a human.
Yeah.
But I think, again, as we mentioned last time, of course, I think it would be very interesting in the future to try to understand peripheral factors that might affect, induced by fasting, as an example, that influence brain autophagy or brain health. Because I think that I certainly considered this previously very much in an organocentric sense. Does fasting affect brain autophagy directly? But it's possible that fasting might do things that increase the levels of various hormones or other diffusible substances that affect the brain indirectly. Do you know what I'm trying to say?
Well, with fasting, I should know this because we work in this area, but I can't think of any papers up. So with fasting, there's an elevation of ketone bodies, right? Beta hydroxybutyrate and acetoacetate, and they serve as a fuel for neurons. But there's accumulating evidence. We had some of this evidence, for example, that these ketones, they also affect gene expression. We found that the beta hydroxybutyrate will stimulate BDNF gene expression, this neurotrophic factor. Do you know of any papers where someone's looked at autophagic flux with ketone bodies?
So there was a man called Bergamini, sort of a more senior person when I started the field. He did some work on that, yes, and it would be consistent with with what you're proposing. Although I think the point that you make about the BDNF is sort of, I think, quite important, so that you could get beneficial effects in the brain that are maybe autophagy independent, but associated with autophagy in the periphery.
Yeah.
And so I think that, those complexities are things that I think will be interesting challenges to resolve in the future.
Yeah. Now there are, maybe I can get you interested in doing some experiments with beta hydroxybutyrate. So there's been clinical trials by Steve Clinane up in Montreal in patients with MCI, mild cognitive impairment, with what are called, they're essentially precursors to the ketones. And so I gives there's our short chain, like C8 carbon fatty acids. And he found beneficial effects. And now him and also my colleague at NIA is doing a trial in Alzheimer's disease of a ketone ester, which is beta hydroxybutyrate. It's actually developed at Oxford by and my colleague who passed away at Richard Veitch at NIH is ketone ester. And endurance athletes are using this stuff now.
Really?
Yeah. And what's her name? Oh my gosh. I'm terrible on names, David. That's like one of my worst problems. Kieran Clark at Oxford.
Okay.
She's an exercise physiologist. And she found, first in animal studies and then in elite British cyclists, that this ketone ester enhanced their performance. And you'll be interested in this. I don't know if you follow Tour de France, but prior to, I think it was 2012, no British cyclist had ever won the Tour de France. And since then, five times, a British cyclist has won the Tour de France. It's been two different cyclists. Those cyclists were taking the ketone ester before anybody else knew about it.
Okay.
And
very interesting.
Yeah. And anyway, we found in Alzheimer's mouse model 10 years ago, we published the ketone ester ameliorated cognitive deficits and had some impact on the tau pathology. Those are the 3x TGAD mice that we made with Frank Lafleur. Yeah, so yeah, you should get some of the ketone.
I should look at that literature. Yeah, it sounds very interesting.
Yeah. Okay, before we went online today in the podcast, you said it would be good to talk about, we've talked about right now, maybe we can slow aging a little bit by, healthy diet and lifestyle habits, exercise, keeping your brain active. But there are other neurodegeneratives, and then stroke, the same thing. Exercise, don't overeat, don't eat a lot of saturated fat, a lot of sugar. But CTE, you want to talk about chronic traumatic encephalopathy, which you have an interest in now?
So I've been exposed to it a little bit. So this is a condition which is pathologically associated with an abnormal pattern of tau deposition in the brain. And clinically, I'm not an expert on this, but I think there's sort of two types of problems that can result. One is sort of psychological problems people get, or psychiatric problems, they get depressed and suicidal, and that can occur, at a fairly young age. But I think that the repetitive trauma to the brain that occurs in many of these sports, that like rugby or American football or soccer or ice hockey or boxing particularly, those people are almost definitely at increased risk of various neurodegenerative diseases like Parkinson's or ALS. And
in the Muhammad Ali is the example of that in the United States, the
yes,
boxer who, he developed a Parkinson dementia type.
yeah.
I don't think anyone did autopsy on his brain, or I don't know if they did, but probably predict there'd be some tau pathology and yeah.
So my concern is that, there are epidemiological data suggesting that certainly for American football, which is being looked at maybe in the greatest detail, that your risks for Parkinson's disease are increased, etc. And not unexpectedly, for professionals who play for a very long time, the risks are greater than amateurs.
Yeah.
But even the amateurs and people who play sort of and finish playing at the end of high school have increased risk.
Yeah.
And so what I'm worried about is that there are many sports in this category. I mean, American football, clearly boxing's, at the edge of the spectrum. But American football's pretty severe. Ice hockey's pretty severe. And then, rugby's, so I'm going to say rugby union, as they'd call it in the UK. Soccer, they did a study where they found that for professionals playing in the Premier League in the UK, that the risks for neurodegenerative diseases were significantly elevated among professional players, except for the goalkeepers who had the risks of the controls.
Huh.
And so my concern is that, there's one discussion about the professionals who have an increased risk of dementia or various neurodegenerative diseases. But there's an equally important discussion to have about the general population, where there are many people who play these sports at school. And even though their risks might be much lower, if they are significantly elevated, and I don't think the data are out there to know what the real numbers are, okay, for many of these sports. For American football, there's a feeling for what that risk is, for Parkinson's, for instance. But they're not huge studies. But what I'm saying is that if the risks are significantly elevated, but those significantly elevated risks are multiplied across a very large percentage of the general population, then the impact from a public health perspective is huge.
Yeah.
And if it's doing something like reducing the age of onset of Parkinson's or Alzheimer's disease by 5 years, the impact of that on a society is just tremendous.
Yeah.
And I think that this is a field where people like Amiki have rarely been pioneers, but I think there needs to be much greater support for people working in that space, trying to answer the critical questions, because, I think that there's certain sports like boxing that we should try to get, banned. I think that it's unethical to have people doing a sport where they're going to actually, the purpose of the sport is to give them brain damage that's going to almost certainly increase their risk of dementia.
Well, and they start, as you mentioned, they start these sports when they're young, and now they have this, I don't know, cage fighting. It's essentially like street brawling, right? Or they kick and punch. And when you're young, and I think we're all guilty of this. When you're young, you're invincible, right? I used to race motorcycles, dirt bike. And yeah, so, yeah, but you're right. I mean, so the studies, it's hard to do in humans, right? Because you've got I guess one way would be like an epidemiological thing where you take older people that did or did not develop Alzheimer's or did or did not develop Parkinson's. And then I don't know how you just have to do it by recollection of relatives of the person, or if they can, the person themselves. And, did that person play football or rugby in high school or hockey? And then, but you have to have huge numbers. You have to have huge numbers of people.
So, Mark, there's another scientific question here. it's a scientific stroke public health question. Sometimes, I mean, I was frustrated during the COVID epidemic where people wanted proper data for everything. Sometimes you don't need proper data. Sometimes you know what the answer is, and the absolute number doesn't make that much difference. So
like masks is like the
yeah, exactly.
There was a
mask was the
controversy
example.
where, I mean, obviously the max, you know, N95 HEPA, essentially you have a HEPA filter over your face. Of course, that's going to reduce your risk.
So you don't need to do the study. I mean, you knew that that was going to have a benefit. And so here the argument is, you know, what level of risk is sufficient to cause one to ask for either ban a sport or change the rules of the sport. And I think
essentially what they're saying is, of course, if you get hit in the head, it's not good for your head.
So for boxing, I think it's a no-brainer. Okay, I mean, I might have my kneecaps broken by somebody for saying that, but I think it's a no-brainer. But then for these more commonly played sports, I think one's really got to think about it hard. And you know, this issue of lowish risk but very broad exposure, and the impact of that is huge. And I think if one's conclusion was there's significantly elevated risk, let's say it's a 20% elevated risk, but it affects half the population, that is worth doing something about. Because you can change lives, you can change health systems. And, you know, after all, these are just games. You can change the rules of the games and you can play another game. You know, you can play basketball, or you can play baseball, or you run.
You know, I heard somewhere recently on the news that, I don't know if it's this next Olympics but flag football, you know, where that you have a little flag on your belt. And so you're not hitting and tackling, you're just pulling the flag off. So that's one thing, you know, but except for the hitting, you know, all the rules are the same as regular football. Hockey, you know, the women, and at least in high school. So I grew up in Minnesota, which is a big hockey state, and I played a little hockey. I wasn't very good, which maybe that's a good thing. But the boys can check. They can, you know, make, you know, physically hit the other player, but the girls can't, women can't. So there's, you know, no checking. You can't purposely jam the person into the boards, or, you know, so there are rules changes that they really don't take out the strategy. The only thing they take out is, you know, hitting. But it turns out for some reason, a lot of people like to see that stuff. I don't know.
I think you're right. I think we selected for enjoying gladiatorial sports, but
I mean, we evolved that way.
Yeah, but I think that you know, now we've got to think very carefully about the real costs. And I don't only mean I really don't mean financial costs. That's a small part of the overall costs of neurodegeneration in our society. So, you know, I just think that people say, have we got cures for neurodegenerative diseases? Well, by stopping people boxing, you can halve their risk of neurodegeneration, at least as amateurs. Okay, not professionals, much higher. You can do much better than that. So, you know, if I said to you, Mark, I've got a subset of the population and I can halve their risk of getting neurodegeneration, you say, you're wonderful. You've done wonderful research. But I've done no research. I've just read a few papers. And I just think that, you know, when we think about neurodegeneration, we've got to also think of what is readily preventable, where we can make an impact. And we shouldn't necessarily ask in any public health setting for absolute numbers if common sense and the balance of risk and benefits argue that a change in the rules could have a significant impact on society.
And then, and in the case of people who are unfortunate enough to inherit a, mutant Huntington gene, or, mutant, whatever, Parkin or tau. So for them, yeah, so these diseases, at least in mice, you can essentially cure them or prevent them from getting it by doing gene editing, or essentially gene therapy, right?
Yes,
You can
reducing the levels of the in fact, with motor neuron disease, you mentioned the superoxide dismutase. Tim Miller's, and colleagues have done, had a very nice trial in people where they showed with that particular mutant protein that they could really impact the course of the disease beneficially by reducing the levels with the gene therapy type approach.
And there's an ethical issue, but it is possible for people to know, like, say, they have a parent who had Huntington's disease, and so they know they have a 50% chance of having Huntington's, and they could, before they marry and have kids, they could get genetic testing to know. And if they want, they could, it's possible with the technology now, they could have a child that they know will not get Huntington's disease through in vitro fertilization, embryo selection. So, the father's sperm, the female's egg, so it'll be the same genetics, except they don't have the mutant Huntington. That can be done now. Because these, at least the dominantly inherited disease, in theory.
Sure, you can just select, yeah, sure.
Yeah. So in theory, with education, but some people have an issue with this, the religious people. But I guess the point is, these dominantly inherited disease, whether it's early onset Alzheimer's, Parkinson's, ALS, and Huntington's, it could be eliminated in one generation.
Yeah, the only caveat to that is that there will be new mutations, but you'll get rid of most of the cases. Pretty much most of the cases, the great majority.
Yeah.
Yeah.
So the reason I mentioned that is that, people with those mutations, it doesn't matter if they play football or, they're gonna get the disease. Unfortunate. So for them, we have to think about other approaches.
Yeah, I mean, you raise another very interesting sort of, people are doing that to some extent for known Huntington's patients, but it raises some very interesting subsequent questions about how one exploits genetics to,
that's true,
benefit subsequent generations, and also some of the ethical issues that are non-trivial.
So as far as preclinical studies with gene editing, a former postdoc of mine, Wenjian Duan, did gene editing in Huntington mutant mice. It had a nice, it was a good journal paper. What about genetic manipulations that enhance autophagy? Is there such a thing? I guess, what would you do? Overexpress
So you could do that, but I think if if you're going to actually go to the trouble of introducing genetic material, why don't you just go for the throat and try to lower the levels of your toxic protein?
Yeah.
So unless you think that in a generic sense, that, for instance, if you argued that inducing autophagy slightly in the brain might improve brain health in the general population, if you get started at an early age, and made you more resistant to polygenic diseases, which actually end up being the biggest problem in our population, it's not a crazy idea, but I'd rather take a safe drug at the moment.
Yeah, I agree with that, and have a healthy lifestyle.
Yeah.
Okay, David. Well, Good. This has been really enjoyable for me. It was great you taking the time. I enjoyed our conversations and hope that viewers and listeners did.
Likewise, and thanks for having me. It was good to see and chat with you.
Yeah. Have a good holiday season over there.
Thank you very much. Cheers, Mark.
Yeah. Bye.